Think Sakshi, where every clue begins with learning. I'm Linda James, your forensic serology educator, and in this series of video, we will be learning about what forensic serology is. And in this particular video, we will be learning about the introduction of forensic serology. So let's get into it. What is forensic serology? Forensic serology is actually a part of forensic science that actually looks for and tests for fluids like blood, semen, saliva, and many more. Sometimes even sweat can be a biological fluid. So actually, it actually answers many questions in a criminal investigation. It actually tells investigator that what kind of biological material is present in a crime scene, whether if it's human or whether if it's animal body fluid, and whether DNA can be identified from it or not. As we all know, if DNA can be identified, then person identification is possible. So, what does forensic serologist actually do? The first and foremost thing that they do is to find the stains. They actually look with naked eye or with flashlights or even some special light sources. They look for the evidences very crucially because finding the body stains is actually the most important thing in forensic serology. Actually, they can also use chemicals that glow or change color in the presence of body fluid. For example, luminol actually produces a fluorescence effect in presence of blood, right? So these kind of chemicals can also be used to detect a body fluid. So to find the stains is the first and foremost thing in forensic serology. Now the second one is the screening of the stain. That means presumptive tests are used. Presumptive test is actually preliminary test. That means the basic test. These tests actually are quick and sensitive tests. These can actually say that it can be or it might be a particular body fluid. Like this can be blood, this can be semen, and this can be saliva, and so on. These tests actually does not confirm the presence of particular body fluid. This actually tells that this probably can be this body fluid. Okay, so these actually are very fast. These tests actually are very fast and can also give false positive results. So yes, as I have mentioned earlier, this cannot confirm the presence of any body fluid. Now we are going to like learn a basic thing about confirmatory test. That means which confirms the presence of body fluid. These tests are actually more specific uh, tests. So these tests are more specific than the preliminary test to say that this is actually blood, this is actually saliva, this is actually semen. So these tests basically confirm the presence of a particular body fluid. Yes. And these confirmatory tests can also check if it's a human body fluid or an animal fluid. Okay. Now, this forensic serologist also preserve the evidence for DNA analysis. As we all know that after this presumptive test and confirmatory test, uh, there is a little chance that these kind of body fluids remain for DNA test. So they preserve the DNA priorly, priorly by knowing that this kind of body fluids will not remain after the presumptive test and confirmatory test. So before testing it with the preliminary test itself, they actually preserve some evidence for the DNA analysis to know whether whose it is, whether if it's a human, animal, and even if it's human, which human holds the body fluid. So person identification is possible by DNA analysis of forensic biological fluids. Okay, now these forensic serologists can also interrupt and report the findings. Like they can combine the lab results with the case context. For example, when the stain was found where the stain was found. They can actually find out where the stain was found, how it might have gotten there, like how did the particular body fluid come to the crime scene. Is it related to the suspect or victim, or some other person has put it intentionally to just mislead the case? These kind of things they can find out. So they can interrupt the findings, like in on the basis of this uh, crime scene contest as well as the lab results. So this can actually help to exclude or include a suspect. So after all this interruption, after all this finding of results, they actually report it. Now we are going to learn about why does this forensic serology matter. Like every other branch of forensic science, forensic serology also helps in linking a suspect and a victim. It also helps to link a suspect and victim to a place, like probably most probably where a crime has happened, that is crime scene, or an object, the weapons that suspect has used, the weapon that victim has been used on, or an event which led to the crime, right? These kind of things we can link with each other. So this forensic serology also actually supports or challenges a story. For example, actually, if a person says that I have never been to this place, for example, that is the crime scene, and if a person claims that he has never been to that crime scene, but forensic serologist has found out his saliva is present on the glass, on a glass cup in the crime scene. So this actually indirectly proves that that person was present in that scene. Whether the person was in the crime scene before the crime has happened or after the crime has happened or during the crime has happened, the statement that he has said that he has never been to this place is actually excluded. So these kind of things can be proven by forensic serology, which is very important actually. So this forensic serology also helps to prioritize to like what to send for a DNA analysis. As we all know that DNA analysis is actually very costly, and every single evidence that we get from a crime scene cannot be sent for DNA analysis. For this, actually, we need to know which evidence is to be prioritized, which evidence is to be sent for DNA analysis, which evidence can actually prove that this DNA belongs to this particular person. So these kind of things should be known before we actually send the molecule for DNA analysis. Now we are going to learn about the history of forensic serology. The history actually begins before 1990s. Like people always suspected the stain at a crime scene could be blood, but they had no scientific way to confirm it. Like earlier tests in 1800s looked at the color of stains. Surprisingly, they actually looked at the color of stains. Like whatever the red thing is, they suspected that it is blood. For example, they often confused this blood with rust, blood with paint, or even blood with fruit juice. So actually, they used to look for the color of the stains because at that time in 1800s, no tests were available, right? All the tests were developed after 1800s. So this actually, these like looking for the color of the stain was totally unreliable. So this unreliability actually led to more scientists to come up with more ideas and developed more tests to help in the forensic serology. So now in 1900s to 1910s, the period of Carl Lindster, who discovered the ABO blood group system. This is actually very good turning point in forensic serology. In this actually, the human blood can be divided into many blood types like A, B, AB, and O. The forensic use of this ABO blood proving system is that, uh, like if in a crime scene, the body fluid of a person, the blood of a person was detected to be type B, and the suspect has blood of type O. So, in this actually, as the blood that we got from a crime scene has type B, and the person that we put in a suspect list has type O blood group, the person in the suspect list can be excluded, right? Actually, he has no role in that crime because his blood is not present. Or like uh, this forensic serology can also act as a tool for exclusion also. Okay. Likewise, if a blood group of type A was present in a crime scene and the suspect is also having type A blood group, then that means that the suspect should be, or can be, or might be the real criminal, right? Yes. Now the pre-DNA serology era. In it actually came from 1920s to 1970s. So more tests actually developed to identify blood and other fluids in this era. Presumptive tests for blood like Benzene test, Kaiser test became more common these days. This actually these tests could even uh, like trace even very small amount of blood, like very small traces of blood can be detected by these tests. Okay, so these are the presented tests for blood. They are like Benzene Kaiser test, which we will be learning in next video, which will be uploaded very soon. So now for semen detection in by the 1930s to 1940s, scientists used microscopic methods like looking for semen cells and chemical markers like acid phosphatase test. These kind of tests actually came into total. Now we'll be learning about saliva detection. These actually saliva detection is actually based on enzymes like amylase, which is actually present in saliva. So, like by seeing all this, in this period, forensic serology was about finding the fluid type and blood group, but it actually didn't prove any individual identity. Like no individual was identified by these methods. Okay, only the fluid type was detected and blood group was detected by ABO blood grouping system. Still, the DNA or person identification was not yet in use. So by this period, like by 1920s to 1970, the courtrooms actually accepted serology as an important part of investigation. So this is a major turning point in forensic serology. Now, in the time period between 1970s to 1980s, forensic serologists actually started using enzyme polymorphism and protein markers in blood and semen. So these actually give more information than just blood type. Uh, but this information doesn't mean that they can actually uniquely identify a person. Okay, so this period is also called as golden age of traditional serology. The period between 1970s to 1980 is known as the golden age of traditional serology. So, in this timeline, the many famous cases used serological evidence to support investigation, but like it doesn't mean that only forensic serology has proved the case because at that time also person identification was not possible, so they actually often needed additional proof along with the forensic serological evidences. Now, DNA profiling came into role by mid 1980s. So, in 1984, Sir Alec Jeffrey actually discovered DNA fingerprinting. DNA testing quickly overtook traditional serology because it could give a unique match. Like in identical twins, the unique match was not possible, or is still not possible. But At that time, this DNA fingerprinting like overtook many other traditional forensic serological methods. But still, even if the DNA fingerprinting was possible, forensic serological, the basic forensic serology remained crucial for the first step. Like, first step means identifying what the stain was. We have already told that finding the stain is more important. I have already mentioned that finding the stain and knowing its importance, knowing what stains should be sent for DNA analysis is much more important. So, detecting blood, semen, saliva, etc., before sending it for DNA is actually important. So, forensic serology still stood strong as before. Even now, it's very important that before DNA analysis can be done. So, traditional serology alone started being used less often in court, but it became the gateway to DNA evidence. So, without the forensic serological identification, DNA analysis couldn't be possible. So, now we are going to learn about modern era, that means 2000s to till now. So, today forensic serology uses a mix of classic test and uh, advanced techniques as well. So, the tests like immunological test, like ABA card test, which you will be learning in like further videos, the RNA and mRNA markers to identify specific body fluids. These kind of tests were actually in use. Are actually in use. Sorry, these are in use till now, till date. So this serology and DNA combination can now solve cold cases also. Cold cases means the cases which are not solved from many years is known as cold cases. So this serology and DNA plays a great role in solving the cold cases and bring justice to the ancient deaths. So it's surprising that, right? It also can be possible. So even actually in high-tech labs also, serology is still the first screening step for body fluids. So now we are going to look on to a case study, the O.J. Simpson trial. This is the name of the case, 1994 to 1995. So in 1994, Nicole Brown Simpson, O.J. Simpson's ex-wife, and her friend Ronald Goldman were actually murdered in Los Angeles. So, O.J. Simpson was a famous American footballer, uh, and he was an actor. He was accused of this crime, like he was accused for murdering his wife and her friend, his wife's friend. So it became one of the most famous criminal trials in the history of forensic serology. Now, what are the important evidence that are got from this case are the blood stains. Blood stains actually were found at multiple locations, like at the crime scene, and that means the at the crime scene, in Nicole's house, and also inside the car of Simpson, like, and also at Simpson's home. So these body fluids were found in these many places. Blood was found in these many places. So serology confirmed the stains were human blood before DNA testing was done. Okay, so now DNA testing after serology. After we got the DNA serological evidence, they actually tested for DNA. DNA test showed that Simpson's blood was found at the crime scene. So this proved that uh, DNA of DNA of the blood that we got from the crime scene matched with the DNA of Simpson. So this actually proves that Simpson was included in this crime scene. Blood matching the victim was found in Simpson's car. So the blood of the victim victims was found in Simpson's car and also in his clothing. So it proves that this actually proves that he has done the uh, crime. So, like in this case study, we can say that this serological test followed by DNA analysis were extremely the strongest evidence linking O.J. Simpson to the murders. However, the defense team challenged the handling of evidence. The defense team, it's the duty of defense team to challenge the prosecution by saying blah blah blah. So they actually said that they claim the contamination and police misconduct. They actually have claimed that police has destroyed the evidence. They have not done the chain of custody very well. So these kind of things were put forward by the defense team. Uh, Despite the scientific results, Simpson was acquitted in the criminal trial, actually, though later found liable in a civil trial. So this case is actually important in forensic serology because it actually shows the power of serology and DNA to connect a suspect to a crime. So this case also has highlighted the importance of proper collection, storage, and chain of custody, which, as we have seen, it was actually claimed by the defense team that proper collection, proper storage, and proper chain of custody was not followed by the police. So this is to be kept in mind when collecting while collecting biological evidence. Um, like by this case, we can also know that even the strongest scientific evidence can also be questioned in a court of law. So everything should be clear from the from our side to know to get a question. This case actually educated the public about forensic serology, forensic science, and made DNA or serology a household term. So this is actually very good case study. Now, what are the challenges in forensic serology? When the biological samples are found in mixed form, this actually can be very challenging for the forensic serologists to identify which are the fluids. So they might be actually confused to use which test, like if blood and semen are mixed together. Like confusion can be there, chaos can be there about what test can be used for detecting the fluids, right? And if, for example, if urine and saliva are mixed, we can't even know what fluids are that, right? So like we all know that in a forensic case or in any case, the fluids are very limited. It's not like we can bring more and more fluids. The fluids are only that much which we can get from the crime scene, right? So these usage of these fluids must be done very wisely. So similarly, degraded and old sample also can be very challenging. Uh, or contaminated samples, like because of various factors like environmental factor or disturbance by animals, disturbance by vehicles or anything. These kind of things can actually destroy evidence. Yes. So limited quantity of sample, as I have told you, in a crime scene we cannot get body fluids in abundant quantity. We can only get the particular amount in which the mm, the particular amount that is actually already present in that crime scene. We cannot reproduce it again and again, right? So wisely using it, forensic biological evidence is necessary. So conclusion, forensic serology actually plays a vital role in crime scene investigation. As we have seen, it can actually link a suspect and victim to a crime scene. And it also involves analysis of multiple body fluids. Like it does not only analyze a single biological fluid. It actually analyzes various kind of biological fluids, even mixed biological fluids also. So it's actually complicated a bit, right? So now presumptive test and confirmatory test are also used in identifying and detecting the body fluids, which you will be learning with me in the next videos, right? So like the advances will continue improving accuracy and speed. So much more development is to be brought to the forensic serological field, which is expected very soon actually. So more developments means more amplification, more reliable and more faster, right? So this is actually crucial for justice delivery system. Yes. Now that's all for today. So if you have liked the video, please like, share and subscribe to Think Sakshi. And stay tuned for my next video about the body fluids. Thank you.